Hi there, everyone. We are so happy to have you, whether you are joining us here today or will be watching from our Cure GABAA YouTube channel. We ask that you please mute your mics if you have not done so already, and include any questions you might have in the chat box below. We will have time to answer the questions at the end of the presentation. My name is Monica Joanna Elnakabe, and I'm the founder and CEO of Cure GABAA, a nonprofit organization with the sole focus to develop potential therapies for GABA-A variants by partnering with world-renowned researchers in GABA-A and the Organization of Patients for Clinical Trials. Today, we welcome Dr. Sarah Poliquin, who recently led our inaugural Cure GABA-A Scientific Roundtable, which was held in conjunction with the American Epilepsy Society. Sarah Poliquin is a distinguished researcher in the field of genetic epilepsies. Sarah holds a PhD completed under the guidance of Dr. Katty Kang, with the focus on unraveling the cellular mechanisms behind genetic epilepsies linked to the GABAergic signaling pathway. During her doctoral studies, Sarah delved into the intricacies of GABAerg2 Q390X mutation, particularly associated with Dravet syndrome. Her groundbreaking work explored the role of protein degradation pathways. Currently, as a postdoctoral researcher at COMBINEDBrain and the Chief Scientific Officer of Cure GABA-A, Sarah remains as an active contributor to the GABAergic disease community. I'm handing it off to you, Sarah. Please share with us the understanding of the GABA-A genes.
All right, thank you, Monica. All right, thank you everyone for joining today. I am going to be giving a brief overview of GABA and GABA-A receptors and their role in disease. So I'm going to start with an overview of how neurons work, and then I will talk about what is GABA, and then GABA-A receptors. And then I will give a broad overview of the known variants in GABA-A receptors. And then I will finish with talking about how you, as families and patients in the community, can help research and the understanding of GABA-A receptors and even your own variant. So as many of you may know, neurons are the main cell in the brain. They pass signals back and forth to each other and talk to each other, and are responsible for everything that our brains and bodies do. So that's what enables us to talk to each other. That's how we move. It controls breathing, digestion, all of that. So that's all through the neurons. And the neurons talk to each other with electric signals. And these signals are passed from one neuron to another at what is called a synapse, which we will be diving more into later. So these electric signals that go from one neuron to another are called action potentials. And these action potentials carry a lot of information, and that is how everything in the brain works, is based on these electric signals called action potentials. And so what they do is there are actually two different, you can have depolarizing and hyperpolarizing signals. So to get a full action potential, you need multiple of these depolarizing ones in red to all join together to reach this threshold to create the action potential to pass the signal along. But you can also have something called a hyperpolarizing potential, where that basically is the stop sign that puts the brakes on things. It will prevent it'll prevent an action potential. It'll prevent that information from going from one neuron to another. And so all of this together is crucial to be in balance in the brain. If there is an imbalance, or if anything is going wrong, then you can get this like huge traffic jam, because just you have the stop and go signals are just not being properly balanced. These action potentials go from one neuron to another via neurotransmitters. So what we have here is an image of a synapse. And so there's one neuron on the left here and a neuron on the right, and then it's zoomed in right here where they're touching at the synapse. And so these little red circles here are representing a neurotransmitter that is being released from neuron A over to neuron B. So these neurotransmitters are small chemicals that act as messengers to help carry that information from the action potential. And then when those pass, then you get the electric signal. So GABA is special. It is an inhibitory neurotransmitter, meaning that it is the blue here. It's one of the things putting the brakes on the system. It's helping to cancel out all of those positive excitatory signals. And this is vital because without GABA putting the brakes on things, the system can just be going nonstop. And that's part of how you can end up with epilepsy and seizures. And so here on the left is just a image of what GABA looks like. So this is a pretty small molecule. So that was a very brief overview of how neurons work and what GABA is. So next I'm going to talk more in detail about specifically GABAA receptors. So in order for GABA to function as an inhibitory neurotransmitter, it needs receptors. And it acts on GABA-A and GABA-B receptors, but for this talk, we're going to focus on GABA-A. So this here is a diagram of GABA-A receptor, and you can see that it is made up of 5 different components that are assembling in kind of this circle together. And so there are 2 alpha subunits and 2 beta subunits and 1 gamma. This gamma can also be a delta, epsilon, theta, or pi, but typically what we see is alpha, beta, gamma. And so the way this works is that the GABA-A receptors are like doors that require a key. So the door is locked until GABA comes along and acts as the key that opens that lock. And then when the door is opened, it allows chloride ions to pass through the door. And so it is the chloride ions that actually are carrying the electrical signal for action potentials or for canceling out action potentials. And in scientific terms, this is what's called a ligand-gated ion channel. So, we have the door here where this is the ions going through. So it's a channel for ions. And then GABA here is just called the ligand here. So this is another view of the GABA-A receptor. And so again, we have our alpha, beta, gamma. And then when GABA binds and opens the channel, then it allows chloride through. So you can maybe get a general idea from looking at the picture how at the bottom here, this can be constricted so that nothing passes through. But then when it opens, it allows chloride through. So when GABA binds here, that is controlling whether the bottom here is pinched shut or whether it'll open to let chloride through. Then when we have mutations in any of these subunits, there are multiple different things that could happen. One of those is that the mutations could change the size of the door, metaphorically. So if you have a mutation that allows more chloride to go through, then that's essentially like having a larger door where you've got all of these ions all just rushing through. And so that would be considered a gain of function mutation. On the other hand, you could have a mutation that makes it so that less chloride is able to pass through the channel. And so then this is a loss of function. Another potential change that you can have due to variance in the GABA-A receptors is they can change the sensitivity of the lock. You might have a mutation that makes it so instead of GABA easily opening the channel you could have like this really secure door where it just doesn't really want to respond to GABA so easily. And so then it doesn't matter how big or small the door is, if the door just kind of generally stays shut and only very rarely opens after you use 10 different keys on it and have to really like yank on it. On the other hand, you could have just an automatic door where you just kind of need to be in the general vicinity and wave at it, and the door opens right up. And so here the sensitivity to GABA is greatly increased. So this would be a gain of function. And so, and you can have mix and match of mutations that change the sensitivity of the lock or of the size of the door. So you might have something where it's very sensitive and is like the automatic door, but yet it also was a tiny little door that's opening. And so this would be a mixed change in the function of the mutation. So you can see how by altering this, it would change action potentials. It would make the shape of the blue line here change. It might make this all compressed and narrow, or it could make that blue line huge. And so it's going to hyperpolarize the cell even more than it is supposed to. And so again, by having an imbalance in these signals, or even just kind of a mistiming of the stop and the go, you disrupt the flow of information from the brain. All of those action potentials traveling from neuron to neuron to neuron get interrupted, or they're all coming at the same time, and you have this traffic jam. There are many different subunits of the GABAA receptor, as I alluded to earlier. So there are 6 different types of alpha subunits, and so all of these are related. And then there are 3 beta, 3 gamma, there's 1 delta, there's an epsilon subunit, pi, theta, and then there are also 3 rho subunits. So all of these are like building blocks. It's like Legos that are assembled together. And even though they are related, there are still crucial differences. It's like, if you have a Lego creation and you can't find the exact piece you're looking for, you can find a similar one and swap it out, and it might work about how you want it to, but your Lego creation is going to be a little bit different if you make any changes. Things might fit together differently, or even just the colors don't look right. And, just all of these have very unique purposes. There's a reason that there are 19 separate ones. And they all have different properties. So this here is just trying to give an overview of how those different subunits are important in the brain. So this here, you can see just that there are the different shapes. So this is the flow of the chloride ions through that channel, through the door. And so here you can tell that like initially there was the huge rush of chloride through the channel, and then it slows down. And so only a trickle of chloride is getting through there. Well, with this particular receptor, you can see how this the stream of chloride through the receptor is staying about the same no matter how long the door is open. And so without getting into specifics of how that works, you can just see that each of these different combinations of subunits will have just kind of different patterns to how the door opens and closes and how much chloride it's letting through. The subunits are also important for for determining where the receptor goes in the cell. So some of the subunits will align directly across the synapse, as you can see here, but some other subunits, such as delta, will generally result in a receptor that is a little bit further away in its distance from the synapse itself. This is called an extrasynaptic receptor. And so synaptic versus extrasynaptic receptors have different properties. Again, they're responding differently to GABA, and they have different roles in how exactly they are modulating action potentials. So we've talked about how neurons work and what GABA is, and then talked about the GABA-A receptor, just how it would work in a healthy condition. So next I'm going to give an overview on variance in the all of the different subunits. This is going to be just quick overview. The slides are a little bit more for reference for later on. I'm not going to be able to go through everything all at once, as you can see here. So this is just some of the various publications that have looked at, in this case, this is alpha-1. So you can see that there have been quite a number of publications identifying a whole bunch of different variants and that they are associated with different phenotypes. So there's epilepsy, developmental delay, hypotonia, movement disorders can result from mutations in alpha-1. There can be micro and macrocephaly, autism, a whole bunch of different things. And then this here, Clinvar, is a database where scientists will put in the different mutations and variants that they have seen. And so what this is saying is that there are a lot of different variants that are classified as pathogenic. Many of you have probably seen on your or your child's genetic report that it may have said that your variant is classified as pathogenic. And so what that means is that this particular variant is changing the function of the subunit, and so that then causes the changes in the receptor as a whole and disrupting the flow of information through the brain. Over here, the VUS, that stands for variant of uncertain significance. That means for this variant, we don't really know what it's doing yet. If it's something that has really no effect at all, or if it's such a minor change where it doesn't really matter. For a lot of genes, there are a huge number of VUSs because there just hasn't been the time and resources for scientists to analyze each and every one of these different variants. But you can kind of tell from the number of VUSs that are being listed how much interest there is in the gene in regards to human phenotype and disease. So with alpha-1, with over 200 VUSs and 55 pathogenic variants, then there is a lot of interest here and a lot of variants are being studied. And Clinvar also shows that some of the variants that are for now classified as benign are still associated with epilepsy. Alpha-2, however, is and has not been associated to quite the same extent with epilepsy and developmental delay. There have been only a handful of papers, each studying only a few variants. And it's noteworthy that I am not putting all of the papers that might be there on a variant on these slides. That would be a lot of information. I'm trying to give just a handful as kind of reference so that you can see just in general that alpha-1 has a lot of research, a lot of variants, and then alpha-2 has less, and so you'll be able to just kind of gauge in comparison. So there are some papers that just for the sake of simplicity are not necessarily being included. But alpha-2, we have epilepsy and developmental delay are some of the big ones. Again, autism comes up. Alpha-3 is much more rare to be associated with neurological problems. There, I have here only one paper, and then in Clinvar, you can see 3 pathogenic variants and 11 VUSs. That is in contrast to alpha-1, where it was 55 known pathogenic variants and over 200 VUSs. So alpha-3 is just less studied in this context, and fewer people with mutations in alpha-3 have been identified so far. Alpha-4 is even more rare. And so this is not to say that alpha-4 variants do not cause neurological problems. It just means that so far, not very many have been identified yet. That could absolutely change in coming years. Alpha 5 is kind of in between with a reasonable number of variants discovered. These are associated with autism and DEEs, so that's developmental and epileptic encephalopathy, a pretty severe phenotype where it's a combination of seizures and developmental delay. Alpha 6, like alpha 4, is far less associated with neurological phenotypes here, just the one paper that was worth mentioning. So that was the alpha subunits here. So then next I'm going to talk about beta, the ones in blue. So for beta 1, there are a number of mutations that have been identified. These result in epilepsy, developmental delay, hypotonia, like many of the others, movement disorder, microcephaly. Only a handful of known pathogenic variants and a handful of VUSs in Clinvar. Beta 2 has much more associated with it. So several of these publications were looking at a large number of variants, too many for me to list out at once on the screen here. There are 41 known pathogenic variants in Clinvar. And then for beta 3, what's noteworthy here is that these mutations can be classified as some of them are loss of function and others are gain of function. And so this is a pretty important deal for beta 3. And if you think back to the analogy of the door, knowing whether the door is too big or if it opens too easily and too quickly is very different than whether, if it's a door that is too small. So that's for the beta subunits, and then moving on to gamma. Gamma 1 only has a very small number of variants known to be causing disease. This was associated with epilepsy, developmental delay, hypotonia. Gamma 2, however, is a pretty common gene to have mutations in. So this is only a handful of the papers out there. There are so many on gamma 2. You see that they cause a range of phenotypes. Epilepsy, which is sometimes mild epilepsy, sometimes much more severe. Developmental delay is common, movement disorders, behavioral issues, vision problems, all sorts of things. And you can see here 85 pathogenic variants in Clinvar and over 200 VUSs. So again, this is a gene that is very commonly associated with epilepsy and neurological problems, and it has received a lot of attention and study. For gamma 3, there is only one pathogenic variant listed in Clinvar, and I actually was not able to find any publications reporting on patients anywhere in the literature. So that's the main ones, but as you can see here, the place of the gamma subunit can be taken with some of these other subunits. So next we're going to talk about these rarer ones. So for delta, that is an interesting subunit because that is associated with the extrasynaptic receptors. So these act a little bit differently than the synaptic receptors. Here there are, again, gain and loss of function mutations that have been identified. And these mutations are associated with autism, epilepsy, developmental delay, like many of the others. Then for epsilon, there are a handful of mutations and papers that have been studied. Also epilepsy with different phenotypes, febrile seizures and infantile spasms, DEEs, which is again developmental and epileptic encephalopathy. Then for the other GABAA receptor subunit, the pi and theta subunits, those are not yet known to be associated with epilepsy or neurodevelopmental disorders. But as I've said earlier, that could change there. And if one of you has a variant in one of those subunits, then the fact that I am not showing that here is not meant to diminish that in any way. It was just that I did not see anything published yet on that variant in PubMed. There are also what are known as the GABAA receptors, which were formerly called the GABA-C receptors. And so these are formed of 3 different subunits, row 1, 2, and 3. And as you can maybe guess from the name that they used to be called GABA-C instead of GABAA receptors, these are pretty different than the GABAA receptors formed of any of the other subunits. The Rho receptors are primarily found in the retina of the eye rather than being in the brain and in neurons. These have not yet been associated with epilepsy or neurodevelopmental disorders. And they form generally a receptor of Rho subunits without assembling with alpha, beta, gamma, like all of these here. So that's the overview of how those work. And so next I'm going to talk about how you can help with understanding the science here. So if you didn't see your child's variant listed on any of these slides, you can help by providing your child's genetic report on Matrix, which is the patient registry that Cure GABAA Variance is using. And if you're confused by all the symptoms, you're like, why does this variant have autism and that one didn't? Why does this one have mild epilepsy but it has a movement disorder or that one doesn't? Then by providing more information about your child's symptoms on Matrix and by providing your medical records to Citizen, this allows us to have just a larger database to understand more about the different variants and if there are any patterns to how some variants might be associated with a particular type of cluster of symptoms while other variants will be different. And then how do I know if a variant is gain or loss of function? So the best way in order to know that is for scientists to study that in the lab. However, that takes time and resources and money and can be difficult to determine large numbers for large numbers of variants. But we are working on what is known as a biomarker. And so this would be something that makes it easier to tell if something is a gain or loss of function. And so for that, you can donate blood and urine to the COMBINEDBrain biorepository. And then researchers working with Cure GABAA can access all of those samples from the biorepository and study those. And so that would make it easier to have what might be called a high throughput assessment to more easily and quickly screen for whether a patient is gain or loss of function instead of needing to wait months for thorough research to be done in the lab. So we have this international community. This is a map here from the community on Matrix. And so we have power in numbers. The more people who sign up and participate, then the more information that the foundation has, the more scientists have access to, and having all of the biospecimens from the biorepository also helps research. So with that, I will take any questions.
Great. Sarah, actually, thank you so much for mentioning Matrix, Citizen, and COMBINEDBrain because our community is, as you know, currently onboarding. And if there's anyone out there who needs any assistance onboarding to any of those platforms, I'll include Augustina's email below, and she can help you get onboarded and get organized. I also wanted to thank you so much for explaining GABA and GABAA receptors. The talk you gave was so simple to understand. And sometimes we get caught up in these big words that us, parents really can't understand. So I just wanted to thank you for making that so simple and explaining the importance of the chloride channel. It really helps us understand why it's important for Cure GABA-A to understand how much of our community is gain or loss of function, because that will help us understand the direction of our research roadmap. So, Sarah, I have a bunch of questions here for you. Our first one is from Mia, and she's asking, can we give our kids GABA supplements? If not, why?
So GABA, when it's taken orally, so as a supplement or a pill, then it doesn't reach the brain. There's what's called the blood-brain barrier. And so this is a protective layer of tissue around the brain that kind of closes it off from the rest of the body. And the blood-brain barrier is really important to the brain's health. It helps to prevent any infections that might have be in your bloodstream and gotten to the rest of your body. It prevents those from getting to the brain. And the blood-brain barrier also blocks a lot of chemicals. So like toxins that might be in nature, some of those are not able to penetrate the blood-brain barrier, and so they don't affect the brain. But it does make it harder to develop and find medical possibilities, because that blood-brain barrier does keep out things that might otherwise be helpful. So GABA supplements, unfortunately, do not pass the blood-brain barrier, and so it will not be able to act on the GABAA receptors in your brain.
And this question by Lisa is kind of related to blood-brain barrier, because she's asking about PBA and Ravicti. She wants to know about the trial. How does PBA Ravicti affect the loss of function in GABRA1? And we could also answer that with gamma-2.
Yeah, so what PBA does is it's what's called a chemical chaperone. And so it helps proteins to fold better, and it helps them to move around the cell better. Protein folding is really complicated. Proteins are a whole bunch of beads on a string, and they have to fold up in just the right way and have the right shape in order to function properly. And so if they aren't folding properly, then it can be trapped within the cell or even be degraded. And what you need is a fully folded protein to move through the cell and reach the plasma membrane at the synapse in order to be functional. And so PBA helps proteins to fold and to traffic. And so it's preventing some of that waste, just the proteins that naturally are not being folded properly. PBA is helping to ensure a greater efficiency of that process and get more to the surface. And so for alpha-1 and gamma-2, then if you have a mutation that is decreasing the number of functional receptors, PBA is basically trying to just counteract that by getting more back to the surface of the neuron at that synapse so it can interact with chloride.
And Luke wants to know, what is the difference between synaptic and extrasynaptic?
Yeah, so for that, I think I'm going to try to find the slide again, and then I'll pull that back up on the screen. So here directly across from the synapse, you have the synaptic ones. What this figure doesn't do a good job of is showing the scale at which this is. So the extrasynaptic receptors are generally much further away. So when GABA is being released from this neuron here, then the synaptic receptors are immediately across from that, and so they get a lot of GABA and are able to respond very quickly. The extrasynaptic receptors are further away, and so it's only as GABA just kind of diffuses through the extracellular space and kind of trickles over there that these ones are able to respond. And so because there is generally less GABA in the extrasynaptic regions rather than specifically in the synaptic cleft, then these extrasynaptic receptors need to be much more sensitive to GABA. And so these are more analogous to the automatic door where you just kind of wave at it and it opens, versus the synaptic ones where you need to like fully insert a key and twist it all the way. Extrasynaptic receptors also will stay on longer, and so like the door, instead of snapping shut, it stays open. This is important for how the neurons communicate. These the synaptic ones are the very fast back and forth modulating, like instant communication, while extrasynaptic, because it relies on GABA kind of diffusing further away, are more of a signal for like the ongoing larger view. So if you have signal after signal after signal, and that is all kind of diffusing to the extrasynaptic receptors, then they'll stay on longer and just kind of keep reinforcing that signal to the neuron of like, hey, look at all this GABA. And that is called tonic inhibition. And that's the role of the extrasynaptic receptors.
Here's a question from Shayna. She wants to know, how does a publication get onto PubMed? And then what is the difference between Clinvar and PubMed?
Okay, great questions. Yeah, so PubMed basically automatically, whenever anything is published in a scientific journal, it will be available on PubMed. PubMed is basically like the Google for researchers. And so there's nothing special in order to get it on PubMed per se. It's just after it's been published. So sometimes scientists and clinicians might have information that they just haven't published yet. And so if, for example, like you know that you've been talking to a researcher about your child's variant, and then it's not on PubMed, that could mean that just the researcher doesn't yet have enough data to publish an entire paper. We don't put just kind of little trickles of information here and there. Researchers want to make sure they fully understand a little bit more of the big picture and have something like worth presenting to the entire scientific community. And so it's only after they have a full publication that requires many experiments, and then it gets peer reviewed. And so other scientists who also study the same thing will then look at the paper and be like, yes, this makes sense, or why did you choose that experiment? That just isn't the right thing to do here. And it kind of goes back and forth until all of the scientists are like, okay, this looks good. You were doing all the right things. You had the right controls. Let's publish this. Let's share it with everyone. And then it ends up on PubMed. Clinvar is different, where Clinvar, instead of having papers with all of this data and experiments, is much more of just a list of variants that have been reported. So this will be from genetic sequencing services or from clinicians. They will submit something to Clinvar. And so it's just like, hey, I found this variant and the patient has epilepsy. And they might not even know whether the epilepsy is due to that particular variant or not. That's, for example, the VUSs, the variant of uncertain significance. It's just kind of saying like, hey, look, we found this thing. Someone else needs to determine if these are related or not.
So does that mean if a variant is of unknown significance, there needs to be more research done, like through biosamples in the lab?
So the main way that VUSs get reclassified is through functional data. So that would be a researcher taking that variant and putting it in cells and then doing experiments to, again, going back to the analogy of the door, like trying to see if this variant causes any changes in the behavior of the door of this of the channel. And so if it is something that causes a pretty notable change in how it the receptor is acting compared to normal wild type receptors, then that variant would be classified as pathogenic. If researchers look at this variant, compare it to all of the control ones that they know are normal, and this new variant is still looking pretty much like all the others, then it would be classified generally as benign. And so that's the most surefire way to reclassify something from a VUS. But the biosamples, as you mentioned, Monica, can be very useful because it takes months and lots of work to do all these experiments to like thoroughly characterize a new variant. And as we keep identifying more and more variants, as more people are getting genetic testing, then there's just this backlog where it's just not feasible to do this thorough testing on all of them. With the biosamples, like blood donations, researchers are hoping to find a proxy of the gain or loss of function, something in the blood where everyone with a gain of function variant has this thing in common, and then people with a loss of function have something else in common that is different from the gain of function. And so then when a patient is newly diagnosed with a brand new variant that no one's tested before, and you want to know whether it is gain or loss of function, then scientists could then look at those things they identified in the blood and say like, oh, wow, this is really matching the people with the gain of function. And so that's what they have. And so that is not 100% guaranteed. You would still need the functional data from a lab in order to know for sure. But the biomarkers can be very helpful and can be much faster and allow more patients to get faster answers than waiting on just a handful to be analyzed in the lab.
Now, forgive me if this is a silly question, but you showed a slide with the phenotypes based on the genes. And we found that there was a lot of research done on some genes and not on others. And is it that if we could get our community organized under citizen and matrix and show researchers that there's like a higher prevalence, like a higher number of patients, this would give researchers more drive to study those genes?
Yes, absolutely. So scientists often do want to help patients and advance medical science. And so when they know that a particular gene is heavily associated with disease, let's take alpha-1 and epilepsy, like that has been known for a really long time that alpha-1 can cause epilepsy. And so scientists have been studying that in great detail for a while. A whole bunch of different labs are looking at it. But then when you have a gene that hasn't been so much associated with problems in patients, then not as many people will they aren't as motivated to look into it. Not to say like, oh, there's no point, because there absolutely is a point. These genes are in everyone, and so it's important to know how they function in normal humans, but you lose out a little bit on the people who might want to study a gene related to a disorder. So yes, by having the increased awareness of like alpha-4, for example, if we find that there are actually a whole bunch of people who have variants in alpha-4 and all those people have developmental delay or they all have hypotonia or something, and kind of get that information out there, then a lab that is already studying GABAA receptors might say like, hey, yeah, let's start doing some experiments on alpha-4 as well and see how that fits in.
And also that would be helpful having that data, that information with their symptoms, their phenotypes when it comes time to clinical trial for clinical outcomes, for outcome measures.
Yes, definitely. So I'm glad you brought up outcome measures. So that is how people determine whether a trial is working or not. So an outcome measure is like the test that you'll be doing. And so that might be looking at the number of seizures. An outcome measure might be seeing whether seizures have decreased due to medication or not. Or it might be, based on movement skills. Like if movement disorders and hypotonia are a big problem, then, and not so much seizures, then you don't want a clinical trial to be wasted by focusing on seizures rather than looking at what is actually wrong. And so having this big database of patient information and being able to really see what it is that patients have problems with and like how many of them there are will help to inform which outcome measures should be chosen rather than it being guesswork of like, oh, I mean, typically this causes epilepsy, so let's look at seizures for everyone, when that might not actually be the most appropriate way to determine whether a treatment is working for that.
And it just is, we keep speaking about Citizen and Matrix, but these two platforms really put the power back into the caregiver's hand, because the data that they put in really dictates what is important to them when it comes to an outcome measure. And the more we hear about, and you really touched on that brilliantly, it's known for seizures, but maybe communication is something that we find that is more important amongst our community. So anyway, I wanted to thank you so much, Sarah, for joining us here today and sharing all this information about GABAA receptors in such an easy to understand manner. And also thank you so much for being our Chief Scientific Officer and leading all of our scientific meetings. We have one coming up tomorrow, so we're so excited for that. Thanks, Sarah, and talk to you soon. Bye.
All right, bye.